attending this week's Conservation Science Seminar, both online, those of you that can hear me, and in person here. It's my pleasure to introduce Dr. Jake Walsh. Jake is an aquatic ecologist, broadly speaking, with a lot of experience. Many of you know him well, but he's part currently of a number of fellows that are part of the Presidential Postdoctoral Fellowship Program in our department. And that program is designed to give new scholars the opportunity to become more competitive for faculty positions, tenure track appointments. So following Jake's postdoctoral appointment, our unit, FWCB, has the ability to initiate that process of transitioning from a postdoctoral scholar and researcher into a faculty appointment. Part of that process is departmental review and in the hiring procedure. So Jake's giving 2 seminars. He already gave a teaching seminar this week. This is the second seminar, his research seminar. And that gives us a way to evaluate his skills, his knowledge, his ability, his potential to join the faculty. So your feedback is really important. There are a number of flyers around the room with a QR code that links to a Google form. There will also be an email sent that has that link as well. And we're soliciting your feedback. That's important that we can, the committee can incorporate that into our review. And lastly, next week we'll do the same. We have 2 seminars from Dr. Marissa Milstein, who is another presidential postdoctoral fellow in our department. So you'll see those feedback opportunities both for Jake and for Dr. Milstein. And faculty have the opportunity also to drop in to meet with Jake. Invitations went out via email, and graduate students in the program had the opportunity to meet with both speakers over lunch following their research seminars. That required sign up so we could get you the lunch. So if you missed this week's, take advantage of next week's with Dr. Milstein. Thank you all again. Jake, we look forward to learning about your research.
Awesome. Thanks, Joseph. Am I coming through on the mic? Yeah, that sounds right. Okay, and I think I'm unmuted on Zoom, so I am good to go here. Yeah, so thank you all for being here. This is, like I said in the teaching seminar, this is a good turnout. I like having faces in the room and people to talk to, and I can feed off of your energy. So I appreciate that, especially after a relatively long week here. A fun week, but a long week. And so I wanted to talk a little bit about some of the stuff I've been working on over the last couple of years. So like Joseph mentioned, I'm an aquatic ecologist. And I want to talk about research partnerships that I've had. That's always been an important part of how I do my work. Since grad school, it's a really important part of, for me, staying enthusiastic about what I do involves connecting with the people who might benefit from my work. And so I've been trying to think about ways to incorporate that more formally into my program over the last couple of years. And so I'm excited to chat more about that today. I did want to spend a couple seconds highlighting my daughters here and talking about Lake Mendota in particular. That's a lake that I learned a lot about through about 9 years of PhD and postdoctoral research. And I think when I think about research partnerships, yes, I think about the people that I worked with in Lake Mendota for those 9 years, but I also think of my partnership that I developed with that lake. I got to know that lake really well through data, and I got to know that lake really well through being on it and sampling it, swimming in it, boating in it, bringing my daughters out to it. I got to bring them back. That was this summer for ASLO, just to see the lake and share that lake with them and share them with the lake. And so that relationship, I think, is really important. And I think in a lot of ways, we all have, everything on the slides. We all have things that we think about when we think about lakes. And for me, that's what I think about there. So, and one of the big things that I want to do with Lake Mendota and with all the lakes here in Minnesota that I grew up with, I grew up here in Minnesota, is to keep them healthy. I think a lot of times we just think about, yeah, they're complicated. There's a lot going on there, but generally we just want them to be well. We want them to be doing well. And as a scientist, this was a concept that frustrated me at times. Trying to put numbers to lake health is impossible. You can't do that. And so I want to talk about how this subjective concept has actually come to define a lot of my research approach, because I think there's ways we can navigate this as researchers. I have a couple case studies that I want to dig into that concept. And then I want to talk a little bit about my long-term vision, a couple of the projects I have going on over the next few years. Okay, so getting into lake health. So lakes are central to cultural identity. Humans gravitate towards fresh water because we need it, because there's food there. And they become culturally important to us. And this is especially true here in Minnesota, in the upper Midwest, where we have a lot of lakes. Here I have different pictures of my family. There's my grandma there on the left, my parents. They grew up teaching me about the importance of living by water and getting to know water and to take care of it. And then that's something I've tried to pass on to my kids. So I have a picture of me and my son there, my oldest daughter when she was very young with my wife, and then again, my two daughters there in the bottom right. And so this importance is something that we pass on from one another. I think in Minnesota, many of you are Minnesotans, or you've been here long enough now, you might have a lake in mind that you care quite a bit about, and you want to make sure that it stays healthy over time. So keep that in mind as I get into more of the science pieces here of what we'll be talking about. So what is lake health? I think that's a question that begs a lot of other questions. For me, one of the really obvious ones is who gets to define what lake health is, and then who benefits from a healthy lake, and are those people who benefit from it at the table to define what lake health is? And so there has been some recent work trying to think about this concept of lake health. This Weinmeier study talked about anthropomorphizing lakes. And this is the way that we can understand lake health in the same way that we understand and discuss human health. And so they talk about thermal and circulatory issues. They talk about respiratory issues, metabolic issues, infections like invasive species. I think they put invasive species under poisoning and other harmful disturbances. But you can think of it either way. And so I think this is very helpful for thinking about what lake health is anthropomorphizing lakes. I think we could also take this one step further in helping us recognize lakes as these diverse and unique beings that we care a lot about. Every lake that we care about is probably different from some other person in this room who's thinking of a different lake. It's facing different issues, it works in different ways, and there are different people caring about that lake in different ways. And so this is something I, again, was taught from my family. Each of these pictures is on Lake Superior. Lake Superior is a sacred place for my family. My grandma's a member of the Fond du Lac Band of Lake Superior, Chippewa. It's just south of Superior. And so it's a place that we always find ourselves coming back to. And it's a place that I often think about when I think about my work. But not only is it a sacred place, it's a being. And I was taught that it's a being. And we talk to it as if it's a being. And it's someone that we want to take care of. And it's someone who's been very important for my family for generations. And I think in a lot of ways, as Minnesotans, we share that with different lakes in the state. And so I think there's maybe a new analogy here. We could take this anthropomorphization of lake health as human health one step further and think about more individualized medicine. I know with genome sequencing, we are at the cutting edge of human health as individualized medicine. I think we have a lot of understanding of lakes, and we have a lot of long-term relationship with lakes that allows us to do this. Maybe more like having a long-term relationship with your family practice doctor. I know when you come in for that first time and you're just another average statistic in a distribution, it can be very challenging, but then you keep going back to that same doctor over time, building that relationship. They get to know you, your body, how it works, what you care about when it comes to your own health. And I think we can do that with lakes. And I think in some lakes, we actually have gotten pretty close to sequencing what makes them tick. Although they're always changing and making that very challenging for us. And so this study also talks about how lake health is grounded in ecological state. So they're talking about things like oxygen saturation, nutrient concentrations, water clarity, and that healthy lakes are those that are close to some natural state. And they go one step further and think about a framework for diagnosing lake health issues and then trying to work towards a healthy lake. And they ask questions sort of towards the end of their paper about more of the community engagement and social connections here. And these are questions that I think as researchers, we still have some work to do thinking about. And again, that's thinking about who defines what aspects of a lake's ecological state are important. Is it clarity? Is it oxygen? What are we talking about here? And then is lake health same from lake to lake? And then ultimately, who's benefiting from a healthy lake? And did they have input developing this model for this Earth's Future paper? I don't know this paper well enough to know if they did or not, but I think it's an important question to ask. There's a really cool management-focused paper out of lake and reservoir management that gets into this monitoring, restoring, protecting, and community engagement framework. And there's a couple things I really love in this lake management framework. One is these loops, this iteration that we keep engaging communities in our work to inform what we're doing and how we're doing it. And again, this is in a management context, but I think you could imagine how research might fit into this. The other thing here is we have this community access in the center that we're always returning to. So I love these 2 points here. And again, my key question here is, where does research fit in this? And in Minnesota, we have a lot of overlap between lake management and lake research communities. And that's something that I get really excited about being here. And I'm excited about some of the partnerships I have going in the state. And so thinking about Minnesota specifically, the Minnesota Department of Natural Resources has this watershed health assessment framework program. They have a great definition of lake health on their webpage. They mentioned this natural state, so thinking about the ecological state of a lake. And then they go one step further here that I like in the definition is thinking about stewardship and recognizing that there is that relationship between people and lakes, and we need to be taking care of lakes and then the watersheds within them. They call it a lake shed here, which I think is a cool concept that ties those 2 things together, land and water. But again, I think that it begs the question of stewardship of what and for who. And when I'm asking these questions, I don't want to set up any sort of straw man argument. It's not like the WAF program doesn't think about these questions. They do. That's a key part of what it means to work at the Department of Natural Resources. But I just want to highlight that these questions are still there, and there's still things we need to chew on as a research and management community. Here's the North American Lake Management Society definition of lake health. And one thing I like that they bring into play here is management of lakes for a wide variety of uses. So we're thinking about, what do we benefit from out of lakes and what are we getting out of lakes when we're thinking about lake health and how we manage them. We're thinking about that they benefit people. And I like the wide variety term there as well, because it recognizes that people value lakes for many different reasons. So when we come back to this question of what is lake health over these few examples, we see that there's an ecological context, that healthy lakes are those that are close to some natural state, whatever that might be. And there's also a social context. Healthy lakes provide a diverse range of services to ideally a diverse group of people. And I think we have a lot of work to do. And I think we all, or not we all, but those of us who are in the lake professional community get really excited about these questions. And I think that this is where I get a lot of my enthusiasm for my own work. Thinking about things like, who defines natural state? What if we haven't measured a lake state? I would say the vast majority of lakes we don't have much data on. And then what if a natural state is no longer achievable? A lot of the disturbances that we've inflicted upon lakes aren't really reversible. But what does that mean for managing in this ecological context? Questions like diverse services for who, and then a really key one is, are all people who benefit from lakes involved in identifying, defining, and investing in services? And then one thing that's valuable for me personally and for some communities here in the state is, are lakes sovereign beings that deserve respect? And then what about the beings within them? I think globally, this is also a very important discussion of what is personhood and who has this sort of sovereignty and agency and identity. And so when I think about it as a key concept of my research program and how I do research, I think about this particular question, and that's, how can we center relationship to define and support lake health? And I mean relationship pretty broadly. I mean that in an ecological context. I mean that in a socio-ecological context. And I also mean that in just the relationships we develop with other researchers, with managers, and with the public in the work that we do. And so I think the first sort of pillar of how I approach this leans heavily on holistic understanding. We need to understand all of those different relationships and connections that are going to be impacting how lakes are managed and whether or not lakes are healthy. Because that is such an incredibly complex undertaking, we need to do this with a certain amount of interdisciplinarity. We need to make sure that we have people from different disciplines doing research and management on lakes, and we need to be bridging silos that may or may not exist in the lake management world. And then finally, what a lot of the focus of this talk is going to be is about partner-driven research. And that is, I think, first and foremost, because research informs conversations about lake health. We provide really important data to provide that ecological context, even to provide that social context of lake health. But we need partners to define what lake health actually means. And we need them there to direct our research to make sure that what we're doing is feeding this more subjective, partner-driven concept of lake health. And I think for me, if we go back to that individualized medicine analogy, I think partner-driven research could be the really big key to that, is leveraging local expertise, local perspectives, and making sure that those loops are all tied together. And of course, speaking of tying all these together, these aren't things that exist set apart from one another. They're all things that feed back, and they're really important to keep in mind how they're connected in how I do my work. Okay, so I have a couple case studies I want to dig into, and I have a couple things that I wanted to highlight just before we get into them. because I think they can teach us about lake health in really interesting ways. So there's different management histories in these two different systems. There are different goals that reflect different dimensions of lake health. And there are different ecological and stakeholder contexts in these. And I think this in particular highlights that need for individualized medicine and research partnerships. So I want to start by talking a little bit about the work that I did through my PhD and through my first postdoc in Madison. And we dug into sort of this uncoupling of different indicators of water quality in Lake Mendota there. And I'm really excited to talk about that because water quality we often think of as a singular concept where everything is aligned. And if you're investing in one thing, you're investing in every other piece of water quality. If you're investing in clarity, you're investing in harmful algal blooms. That wasn't the case in what we were studying here. And so Lake Mendota, that's pictured here in this photo, is a culturally important lake to south central Wisconsin. It forms the northern shore of the Madison Isthmus. Madison is the capital of Wisconsin, and people use it for all sorts of reasons, much like the lakes we have here in our own metro area. They're out there fishing, they're out there boating, swimming, skiing, sailing. Sailing is huge in Madison. It's being used for all kinds of things. And lake health, especially water quality, is highly valued by these diverse stakeholder groups. And lake health has been a conversation that has been at the forefront of the town of Madison and the broader community of South Central Wisconsin for the past 50 plus years. How is our lake doing? And there's a really strong vocabulary around water quality in that area. And one of the key challenges is that, like a lot of our lakes here in the Twin Cities, Lake Mendota is in a highly agricultural and urban watershed, and this leads to high nutrient pollution from runoff from the agricultural and urban watershed into streams, into the lake, and poor water quality as a result. It might be kind of hard to see, but this lake is pretty green. There's a blue-green algae bloom going on out there in this photo. And so my thesis work, I didn't frame it as centering relationship in Lake Mendota at the time. I was just trying to understand all of these different connections and make sure that people were plugged in there in some way, shape, or form as both a cause of what's going on in the lake, but then also people who, as another piece of the system that benefits from the lake itself. And so I sketched all of this out in large part because there is a lot of research on the lake itself, and we have a really good understanding of some of these connections. And so I was able to leverage some of that. And so a lot of the foundational research in Mendota is about this underlying relationship between the watershed and nutrients and water clarity through algae blooms and algal growth. And in particular, there's been a lot of study on the species of zooplankton, Daphnia pelecaria, and it's a particularly large and efficient grazer of algae. And in Lake Mendota, it's actually abundant enough where it can clear the water of algae and improve water clarity as a result. And so not early, I guess it's several decades ago, but some management that happened in the 80s tried to leverage the strength of Daphnia in the system and the importance of Daphnia in this system. And they did that through a biomanipulation of stocking a huge amount of big fish-eating fish to reduce predation pressure on Daphnia, allowing them to grow more abundant and bigger, and clear the water of algae and improve water quality as a result. And this was largely a success. Stocking these big fish-eating fish had these cascading effects on clarity. It boosted the Daphnia population, and then water clarity was much better as a result. Here I have it as a very literal interpretation of a Secchi disk depth. You drop it into the lake, when it disappears, that's your Secchi disk measurement. And so the deeper you go, the clearer the water. So water quality improved with more Daphnia after this management effort. That was until spiny water flea invaded. And we were nervous about this for two reasons. One is spiny water flea preys selectively on Daphnia. Daphnia are big, slow, and juicy. They're an excellent meal for spiny water flea, and they are poorly adapted for running away from spiny water flea. And they were also, spiny water flea was detected in 2009 at some of the highest densities on record. And so our management partners at the Wisconsin Department of Natural Resources, at Dane County, where Madison is located, and then with some of the local lake groups, we're really nervous about what spiny water flea could do to unravel this system where we have good Daphnia and good clarity. And so here's sort of where spiny water flea sits within this system. And you can see they indirectly influence water clarity there through Daphnia. And so one of the first things I did when I was in Madison working on my PhD was thinking about how much does spiny water flea impact Daphnia? How much does it increase predation pressure on Daphnia? And I found that they more than doubled predation pressure on Daphnia in Lake Mendota. And this led to a roughly 60% decline in Daphnia biomass, though some years were much, much worse than that. And so on average, this led to a 1 meter decline in clarity, almost entirely offsetting the efforts of that biomanipulation in the 80s. And like I mentioned, water clarity in particular is extremely valuable to the region. I spent a lot of time thinking about the best ways to communicate with people. I came up with fun little cartoons about seeing your toes in the water. I like to see my toes in the water as much as I am getting more and more comfortable with what lives down there when I'm swimming. And they've also, because there's been a strong research community in Madison for so long, there has been social scientists who have asked questions about how valuable is water quality and what would you pay for improved water quality in Lake Mendota. And in this study, it was gaining 1 meter, and that's what we lost with spiny water fleas. So for my thesis work, I was able to update the numbers from that 2001 study. And what we found was pretty shocking, though not entirely surprising. But that 1 meter of water clarity in this one lake was worth something like $140 million to all of Dane County. So that's what I've highlighted there in the map. So again, a big staggering number, but not surprising given how people use this lake in Madison. And so this story was really easy to plug in. Like I said, the vocabulary around water quality and water clarity in particular was really well established in Madison. And so going out and talking about, oh, we lost 1 meter of clarity, now we have to redouble our efforts to reduce the nutrient load into the lake was a very straightforward message. However, one thing that we found as we kept to dig deeper and deeper into this study is that lake health was much more complicated than water clarity. So here's just a time series. We have 2 things that I'm tracking over time. In the gray lines, we have Secchi disk depth. That's how clear the water is. And then in the sort of black bars with the dots on them, that's the beach days lost to harmful algal blooms. And so we found, yes, water clarity did decline with the spiny water flea invasion. However, so did the beach days lost to harmful algal blooms. We saw better beach conditions, even though clarity went down. And so these 2 indicators of water quality that used to go pretty much hand in hand with each other are now decoupled, which makes for more complicated messaging for sure. And I'll get into that in a second here. And so some of the long-term data that has been collected from Lake Mendota for the past 50 or so years put us in a really great spot to do some time series analyses to think about important connections between Daphnia and algae and nutrients. And so I have 2 communities of algae here that are both influenced by Daphnia. One is cyanobacteria. Those are the ones that lead to harmful algal blooms, and then one are diatoms. So it's a dominant community in Lake Mendota, but they don't lead to harmful algal blooms. And you see here that Daphnia don't seem to control harmful algal blooms in Mendota. And I apologize, this was back before I was more sensitive about color in my figures. But we have a thinner blue line pointing to cyanobacteria, where it's a weak, actually positive effect on cyanobacteria, and then a strong, thick red line from Daphnia pelicaria down to diatoms, a strong negative effect on those algae. And in particular, cyanobacteria, they weren't as strongly controlled by Daphnia. In fact, there's some evidence to say that Daphnia might promote cyanobacteria in the lake. Instead, they were more influenced from the bottom up by nutrients. And we found that nutrients declined with invasion. So phosphorus is the key limiting nutrient in a lot of lakes. It certainly is in Lake Mendota. And you can see that phosphorus over time, measured in different fractions here, we have total phosphorus in orange, dissolved reactive phosphorus in blue with those triangles, and that's the phosphorus that's available to algae for growth. Both declined with spiny water flea invasion. And so I was really interested in this, and I had some sediment cores that I had used for a different research project, and I knew that there was a million different things you can do with sediment cores. I just didn't know what you could do or what would actually make sense. So I partnered with 2 postdocs who were at the UW CFL at the time, Dr. Jess Corman, who's now at Nebraska-Lincoln, and Dr. Sam Munoz, who's now at Northeastern University. And we tried to pair this long-term ecological research data with the paleolimnological record in Lake Mendota to dig into what is going on here. Why are blooms of harmful algae no longer coupled to clarity? And there's a lot going on in this plot. But if you look at the sediment core itself, you can see these bands, these white bands moving through the core. And what we found by coupling that with the long-term ecological research data is that these blooms were likely triggered by big algae blooms. So if you have less Daphnia, more algae blooms, but then these calcite whitings, that changes how the biogeochemistry in the lake is working. And so digging even deeper, and again, apologies for this complicated plot, what we found was that when there was more calcite whiting events or better conditions for those calcite whiting events to occur, there was less phosphorus in the lake. So those of you who are in the lake management community, this is similar to alum treatment. So alum is a powder you put in the lake. It binds to phosphate so that it's no longer available to algae for growth. And then it sinks down to the bottom of the lake like in a goop. And calcite is similar. When calcite precipitates out of the water column, it can bind to phosphate, that bioavailable phosphorus, and sink to the bottom of the lake. And so what we saw is that with more blooms, these whitings were happening more and more often in the lake. And this is actually a negative feedback on eutrophication in the lake, or the lake becoming more and more productive, which is really interesting. So it explained that lower phosphorus. It explained possibly why we were seeing lower clarity, but also fewer harmful algal blooms. And so this story was a little more complicated, but our partners were excited about it. We were talking about the relationships between clarity conditions and beach closures with harmful algal blooms. And then zebra mussels showed up in 2015. I like to joke that they literally erased my entire thesis through their filter feeding in the lake. That's not totally true. We're actually finding that the lake-wide effect in Mendota from zebra mussels is much harder to detect. Mendota is a relatively large and deep lake, so there's only a thin band where zebra mussels might be impacting anyway. But then the other key thing here is I moved on to a different postdoc here at the U, and I wasn't able to iterate back with our partners on, okay, what does this mean? What does this decoupling mean to you? And what does it mean for how we manage this system? So just to summarize really quick here for this particular story, clarity as a consistent definition of lake health, like it was for decades before spiny water fleas showed up, became much more complex. Clarity and harmful algal blooms were decoupled, and that changes how we think about lake health in Lake Mendota. Zebra mussels, we would also expect to have a similar effect. So if they were to have a lake-wide effect, we can see that, or we see in other lakes, that they can often reduce clarity. So I said they're filter feeders. They pull algae out of the water, increase clarity. By pulling algae out of the water clears up. But this also moves nutrients into the shallower parts of the lake, and that can lead to these big benthic algae mats. In the Great Lakes, those can break off, they can rot, they can lead to anoxia in the lake, they can lead to nasty, awful beach conditions as they wash up on shore. And so again, this is a decoupling of these different dimensions of water quality, and it complicates what we're talking about. And I think these complex discussions need to happen in partnership. Otherwise, we're just reporting things that I think are hard to message on, and they're hard to communicate about with the general public or with managers. And so if I had sort of infinite time and infinite possibility, going back and doing this and iterating over this problem in medicine would have been really productive, I think. All right, so the second case study I wanted to talk about today is what I've been working on more over the last year and a half. And that's in a partnership that we have with the Minnehaha Creek Watershed District and their goal to evaluate the effectiveness of common carp management at a statewide level. So here is our research team. This project was funded through the Minnehaha Creek Watershed District. It's in partnership with Dr. Dan Larkin. We had these initial conversations with the Minnehaha Creek Watershed District. I think the first fall that I was here, time is a little bit fuzzy in these sort of later years of like the early part of my career. And we also have partners from the Minnesota Pollution Control Agency and the Department of Natural Resources. And so the key issue here is, of course, common carp. So I say, of course, because this is a group of people who probably hears way too much about common carp, and maybe some of you are even tired of hearing about it. But you hear about it because it's important. It does harm lake health in some cases. It's a widespread non-native fish. Its feeding method dislodges aquatic plants. It stirs up sediments, which can release nutrients that promote algae growth. The sediment itself also reduces water quality just by muddying up the water. And then the key thing about dislodging aquatic plants is that aquatic plants are important for fish habitat, and they're also very important for waterfowl in the state. And management of common carp can reduce these harmful effects. So I'm gonna have a couple plots here from a study from Lake Susan here in Minnesota. This was done by Dr. Shemek Bejer, who's here at the U, and who owns Carp Solutions here in the Twin Cities. And they found in their study that aquatic plants responded positively to removing carp. They pulled carp out of Lake Susan, and the vegetation cover in the lake increased. They were able to grow deeper and spread into deeper waters with the clearer water. And they also found clearer water and lower suspended sediments in the water column. So common carp management can reduce their harmful effects. It can improve lake health. But there's an underlying challenge here, and there is a fair amount of variability in how lakes respond to carp management. These are pictures from Coleman Lake here in the metro area. In the pre-photo, so in 2006, over there on the left, you can see a relatively thin band of water lily all around the lake, but the water is relatively open outside of that. Once they removed carp and clarity improved, the lake became overrun as a value-laden word, but it became much more full of coontail, which is a native plant. However, there's also, and you can't see this, but you kind of see the color, there's also these metaphyton or clumps of algae mats that grow between the coontail plants. And this is written by one of the local, I think Lake Association members, but possibly from the management group that helps manage these lakes. And this is a relatively contentious issue. They got better clarity, which is what they were hoping for, but now they're seeing these native coontail expansion into the lake, which is causing weird algae blooms in the near shore. And again, it's decoupling these two things we used to think were in pretty tight alignment. And this makes for a very complicated discussion about what is common carp management for in terms of lake health. And so these were some of the questions that MCWD had because they were seeing similar things in their lakes in the 6 Mile Creek subwatershed. And so they saw pretty widespread success in reducing common carp biomass in their systems. So in this plot in the before on the left, these warm colors are associated with higher carp biomass. And you can see after, you see more cool colors. So they saw pretty much widespread reductions in common carp biomass. However, the lake responses were pretty variable. And so I'm going to unpack this a little bit. I'm going to show a couple of these sort of arrow figures throughout the talk, so I do want to spend some time on it. Here I have arrows pointing from pre to post. So the end of the arrow is post management. I have a goal of like the direction we want the arrows to go. And so in this case, it's plotting Secchi depth against floristic quality index. They're two measures of lake health that MCWD cares about. With higher clarity, like I've discussed in the past couple examples, we think of healthier lakes with higher clarity. So moving along that axis is a good thing. And then the y-axis is a little more comfortable or complicated. As you move up the axis, lakes get healthier with a higher floristic quality index. And floristic quality bakes in a couple things about a plant community. One is its species richness. If there's a lot of different kinds of plant species in the lake, you're likely will have a higher floristic quality. But it also cares about what kinds of species are in the lake. And it cares about lakes that are sensitive to disturbance, lakes that you might only find in systems that aren't disrupted by nutrient pollution or other human mediated effects. And so moving up this is both a mix of high richness and more highly sensitive species. But again, you can always just think of left to right, healthier, down to up, healthier. And you can see here by looking at changes in Secchi depth and floristic quality in MCWD lakes is that they saw a general increase in floristic quality, which is great. That's what they're shooting for. But the clarity story is much more complicated. You can see that line is more vertical than pointing up to the top right corner of the map. And the other thing is, if you look at the individual lakes themselves, they're pointing in every which direction. These lakes are responding in a huge range of ways. And this is something that MCWD was really interested in digging into. And so we had a couple key questions driving this. One is just trying to establish some of the key ecological relationships in the lake. Is the lake working like we have hypothesized in our minds? And the second is how variable are these lake responses to carp management and what drives this variability? The second one is super important for MCWD in particular because they have these goals of trying to set data-driven expectations for what people should anticipate when doing a carp management project. MCWD has a lot of managers and stakeholders coming to ask them, I want to pull carp out of my lake to improve water quality. And if this is the data they're seeing, there's a fair amount of hand-wringing going into that decision of, man, what are we actually trying to do here? And so the goal here would be to try to provide some insight into the factors that influence management success. Try to figure out what's sending those arrows in different directions. And can we use that to lead to better projects for managing common carp? And so I've talked a lot about this arrow connecting carp to water and floristic quality. But of course, these lakes exist in a relatively complicated management system where we have managers like the MCWD impacting the watershed and trying to influence the watershed to influence water and floristic quality, but also treating the lake directly through alum treatments like I described to reduce nutrients and reduce algae blooms, and then also treating invasive aquatic plants to get a healthier water or macrophyte community. And so our project goals were to apply these questions across the state of Minnesota. We wanted to know, what are the key relationships here? Can we define some of these arrows? And then what are lake responses to common carp management? And what are the drivers of those responses, more importantly? And so we went about this by collecting statewide data. This was led by Jill Sweet at MCWD. She's now a master's student in water resources sciences. Super excited to see what she comes up with for her thesis, working on that with myself and with Dr. Larkin. And so she worked with contractors, managers, and lake associations to pull together all of these different data sets on carp biomass and removal estimates. We also got water quality data from the Minnesota Pollution Control Agency. We leveraged the MACIRC PI Charter database, which is a massive database of point intercept surveys, in large part of efforts from people who've been collecting those point intercept surveys for decades, and then the efforts of Dr. Mike Veerhoven, Dr. Dan Larkin, and then where'd Alex go? Dr. Alex Badges, and trying to pull all of those data sets into one place. It is truly an amazing resource, and I will be publishing on it for the next 20 years probably. So thank you. And then we also got lake and watershed management data for the Minnesota Pollution Control Agency. And so to get at our two different questions, we sat down with our partners to think about what might be useful and do these techniques sound like they would be informative for you. So to quantify relationships, we settled on structural equation models. They gave us a couple advantages. One, they can help us quantify those arrows. What is affecting what in this complex system? And then two, it allowed for a relatively messy and patchily available data set to work well. Using piecewise structural equation models, you can actually draw on different sets of information depending on what variable you're going at. So when I talk about those models, one, know that that's a cool advantage of them, but two, it's also worth keeping that in the back of your mind as you're interpreting it, is that it's not a single set of lakes for each variable. It's a different set of lakes for each variable. And I'll probably just want to remind you of that when I talk about those anyway. And then the other thing for quantifying lake responses, we actually settled on a pretty simple approach of just looking at change before and after a removal. Removals are somewhat of a gray area where you might remove for a single year or a single season and see the response you're looking for, or you might need to remove for a few years. And so we worked with MCWD, we worked with local managers to think about, here's your project, where would you draw the line for before and after a removal? And so there is a little bit of decision-making going into what before and after is. And so here's sort of a hypothetical model, a simplified hypothetical model of what our system looks like. We have different relationships connecting all of these pieces that we're paying attention to. And we might hypothesize that pulling carp out of a lake has a negative effect on carp biomass. We know that because of mass conservation or whatever physical law that is. And we also might expect, or not expect, but we hypothesize that there's a negative relationship between carp and macrophytes. So here I have it measured as littoral frequency of occurrence of submersed plants. So that's the coverage of submersed plants in the area within a lake where plants can actually grow. And then we also expect a negative relationship between carp and clarity because they're stirring up sediment. A positive interaction for the same reason between carp and nutrients because they're stirring up sediment. And then we talked a lot about that relationship between nutrients and algae. And here we're measuring algae using chlorophyll A concentrations. And then between algae and clarity, we expect a negative relationship. And then we expect a positive relationship between clarity and macrophytes because plants need light to grow. If there's more light, they can grow into deeper depths. And then there's also some key indirect paths here that structural equation models can help us quantify. And so there's a few things going on. I have them colored in different paths. There's a green arrow that suggests that it's a combination of these two paths. But just to show that carp have negative direct and indirect effects in this system. I think one negative indirect effect that we talk a lot about is this yellow pathway going from carp to nutrients to algae to clarity. So there's both a direct effect on clarity and an indirect effect on clarity. And then because of that, we would expect harvest to also have some indirect effects on some of the things we care about. And we would expect that they would be positive in terms of clarity and macrophyte coverage are measures of lake health. Okay, doing all right. And so talking about some of these structural equation models, I alluded to this a little bit. The key thing here is it provides us with a series of mixed effects linear regression models. They're fairly simple to estimate some of these direct and indirect relationships. And again, remember, we have different amounts of data for clarity and nutrients than we do for like macrophytes. We have slightly less data for macrophytes, but this piecewise structural equation modeling approach allows us to use all of the data to define some of these arrows. Okay, and what we found is that largely our system is working the way we think it is. A lot of the interactions we found are consistent with what we hypothesized. So we're seeing negative effects of harvest on biomass. That's great. We're seeing the positive effects that we expect through this carp to sediment to nutrient pathway, and negative relationships between clarity and algae and positive between, oh, sorry, I have myself here, positive between clarity and macrophytes. And the story is a little more complicated between carp and macrophytes, and we think this is a seasonal issue that we're trying to tease apart now. Macrophyte communities look really different in the early months of the year, so like April through June, than they do from July to September. And so, and one of the big reasons for that is invasive curly leaf pondweed, and that's another key piece of this discussion we have with our partners. So we're trying to dig into that right now. We also were able to estimate some of these indirect effects. Here, I try to have them color-coded because I know it's fairly complicated. I also have the direct effects themselves right next to the indirect effects, just to sort of give you a comparison of magnitude. And these numbers, they are standardized. So we're talking about 1 standard deviation change in a driver leading to 1 standard deviation change in a response variable. So it's a relative measure. And so it allows us to compare apples to apples as we're moving across all of these different units in this system. And so we do see a significant negative effect of carp biomass indirectly through this nutrient algae pathway on clarity. And we see a negative effect indirectly through the 2 different pathways that are indirect and lead up to macrophytes. So maybe we're not seeing a significant direct effect, but we are seeing a significant negative indirect effect through clarity in these 2 different blue and yellow arrows. And thinking about the carp removals themselves, the management that we're trying to do, we saw a significant positive effect of carp removal indirectly on clarity. So again, through the blue and yellow pathways, and then through carp, and then an insignificant effect, though I would say marginally so, on macrophytes themselves. So a couple take homes here. One, generally, this modeling approach supported our hypotheses about lake responses to removals. But one thing that we've found is in some of our post-hoc analyses, our modeling suggests that there are some missing pieces here. And I'm going to get into what I think that is in a second, or why I think it is. But I think it's related to this suspended sediment question. So carp are stirring up sediment, which we were hoping to capture with that direct arrow between carp and clarity. But it might be that there's more going on here. And I'll get into why I think that is in a second. Okay, so in terms of lake responses, so here I just have some histograms. The 0 is no change. I have a line denoting that. So left of the line would be a decrease in the variable we're looking at. Right of the line would be an increase. And then the y-axis is just the number of lakes where we saw that particular change in that variable. So we were hoping to reduce carp biomass. That's the first panel on the left there. And we saw overall a pretty strong reduction in carp biomass on average. But you can see there's a pretty wide range. And that range is largely dependent on the initial carp biomass in the lake, which is kind of what you'd expect. In the middle plot here, we see changing Secchi depth. Again, a positive, relatively strong increase on average, but a huge amount of variation around that, where you have some lakes that are maybe slightly increasing, and then some lakes where the change is relatively small. And then flourish quality increased by 4.8, which is a pretty substantial increase given the actual magnitude of the index we were interested in. But again, we're seeing some variation there. Moving on to a few more variables here, we saw consistent declines in total phosphorus, but again, there's variability in that response among lakes. A very small decline in chlorophyll A. And I might even question if that's biologically relevant, minus 4.5 micrograms per liter. I think a much stronger take home here is the chlorophyll A responses varied. It varies quite a bit around 0 there. And the same story is true for submersed macrophyte cover, where it's no significant change on average, but some lakes are increasing, some lakes are decreasing in terms of the macrophyte cover in them. And so, like I said here, I mentioned that suspended sediment might be some of the key to what's going on here with our structural equation models, and it's not accounted for in our particular approach. And so I think there is a key role of sediment here, and this is why. So here I have a plot of chlorophyll A on the x-axis, Secchi depth on the y-axis, and just to show change in those two variables. You would expect a decrease in chlorophyll A means decrease in algae. You would expect if algae was impacting clarity for that to lead to an increase in clarity. And what we're seeing here is that on average clarity is increasing, but there's a minimal decrease in algae. And so even in those lakes where we see really sort of vertical arrows in the higher Secchi depth points on the far left there, they're not changing much in terms of their chlorophyll A. And so I think those increases in clarity where chlorophyll A isn't decreasing could be due to suspended sediments. And so that's, I think, a big question mark that we'll have moving forward, given it wasn't a variable that was broadly available for our lakes. Okay, so just to wrap up this CARP piece, and I'll try to keep moving here. We saw variable responses among lakes. We saw some diverging, especially in like Coleman Lake, we saw diverging dimensions of lake health where clarity and floristic quality, or clarity and macrophyte coverage were coming into conflict with one another in terms of stakeholder perceptions. And then we also saw that these are generally what we found was consistent with our hypotheses going in. There could be this interesting role of sediment impacting clarity. And we still need to explore the drivers of variability in lake response. And that's what we'll be doing over the next 2 months as we wrap up the project. Okay, so I wanted to circle back on both of these because I think there's some similarities that both cases teach us. And so first is this concept of individualized medicine. I think both of these show that lake health is context dependent. We were talking about different things in both systems. There's different ecology going on in both systems, and people are perceiving things differently in both systems. And there's also a lot of different perceptions and perspectives within a system, which is an interesting conversation. And that different dimensions of health are not always aligned. We saw clarity and harmful algal blooms decoupling in Lake Mendota, and then we saw clarity and macrophyte coverage somewhat decoupling in the carp lakes. And I think researchers can help us define how we prioritize these different dimensions, given that they're not on that same aligned track that we thought they were. And I think another thing here that's really valuable, one thing that I'm excited to keep doing with carp management here in Minnesota is to continue iterating through this process and trying to refine the definition of lake health as it is informed by our research and by our partner goals. Okay, so I have some time to talk about some projects and my long-term vision here. And I'm excited about this. I think I was able to secure some funding in a few different projects. I'm able to move forward on some of the CARP stuff that we're doing, and that's thanks to the Macerk Labs to Lakes initiative. We'll be helping evaluate lake responses in the 2 study systems that they're removing CARP for this initiative. And so we have partnerships with the Sherburne County Soil and Water Conservation District. We have partnerships with a partnership with the Chisago Lake Chain of Lakes LID, Lake Improvement District, and then also multiple Macerk teams, including Dr. Solomon David and the GAR Lab. They're evaluating native fish responses for this project. I'll be bringing in a master's student for this project to help with collecting and analyzing our data. And then our goal, like I mentioned, is to evaluate the lake responses in these systems to carp removal. It'll be very similar to our MCWD work, and I think there'll be really good complementarity there in these 2 projects in thinking about water quality and the macrophyte community. I'm hoping to go back to my zooplankton roots a little bit here and collect some zooplankton data. And that's for 2 reasons. One is there's a carp removal project in Madison that shows some really interesting zooplankton responses to carp removal. And 2, it fits in really nicely with the food web analysis that the GAR Lab is working on. So Dr. Solomon David. Third, I've been working over the last, this is a different point. So next, I've been working really hard over the last 2 months thinking about sedimentation dynamics in these lakes. This is a big question mark in our MCWD project, and I've been talking a lot with the St. Croix Watershed Research Station about what kind of coring work I would need to do to really capture what's going on here. And I'm excited to get back into paleolimnology. The work is hard, but the data is just spectacular, and I think it's going to be really informative. The other project that I'll have going in my lab is supporting innovative and adaptive invasive aquatic plant management in Minnesota. This is where I spent the vast majority of my time working at the DNR for 3 years before I came here for this postdoc, was thinking about how do we manage invasive aquatic plants. I spent half my time managing a grant program for that, and the other half of my time trying to coordinate and understand research needs in our program for invasive aquatic plant management. And so I get to partner back with my DNR colleagues on this one. Very excited about that, about working with people that I really love to work with. I'll be bringing in a master's student and a research technician for this project. And we're going to be really focusing on data harmonization and sustainability. There's some really some low-hanging fruit and opportunity using DNR permitting data that they have pretty well organized internally to think about how that impacts, how management might impact aquatic plants generally. And then we're also trying to think about how do we close the loop on some of the treatment data. Pesticide application records, so that third bullet there in particular, they're mandated to be recorded alongside of treatments. And contractors need to hold on to that for 5 years, but then it goes away. There's no loop where it comes back to the DNR or it comes back to researchers. But I think they're in the contractor community, there's a fair amount of interest in trying to close that loop so we can do better and more adaptive invasive aquatic plant management. And so we're hoping to get them together and think about what are the possibilities there. And then a key part that the master's student will be working on is trying to evaluate invasive aquatic plant management effectiveness. There's a lot of great work from Dr. Mike Vierhoven on curly leaf pondweed management that we could help to inform if we have more data. But I think there are other species that will be interesting to investigate for effectiveness. But the really big question that I'm interested in here is this uncertainty question. Where are the big question marks in our data set and our understanding of how management influences aquatic plants? And how can we reduce that uncertainty over time? And how can we use our analyses to inform monitoring and experimentation that might reduce that uncertainty over time? So really leaning into this adaptive piece of our project. The last project that I want to talk about, I am extremely excited about. This is what I had in mind when I was like pitching myself as a researcher on the job market 5 years ago or 6 years ago, is I wanted to go do awesome long-term zooplankton work in Minnesota. We have great long-term data in Wisconsin, where I did my PhD work and my first postdoc work. And we have fantastic zooplankton data and long-term lake data here in the state. And so this one is also in partnership with the DNR. We're working with the state zooplankton specialist, Kylie Couture, and a fisheries researcher, Dr. Heidi Rantala, 2 people I've worked with a lot in the past through Dr. Hansen, and really excited to work with them on this project. And we'll bring in a PhD student for this project as well. And the goal here is to leverage this really great long-term data through 3 programs, one being the Large Lakes program. Those are 11 of the large walleye lakes in the state that we monitor very well for purposes of sustaining great walleye fisheries. And then there's a couple dozen sentinel lakes, and those lakes were selected to be a representative sample of all the lakes in the state. So it's our way of keeping our finger on the pulse of how lakes are doing in the state. And each of those data sets have really great zooplankton data. And we're hoping to quantify the role of zooplankton in provisioning ecosystem services. So I've talked a lot about the relationship between zooplankton like Daphnia and Clarity. We're interested in that piece of that in the state of Minnesota. But then we're also thinking about how do zooplankton support different fisheries in the state as well. One thing that I am especially excited about is using zooplankton as a biological indicator of lake health. So the Minnesota DNR has their Watershed Health Assessment Framework program. They use these biological indices to give us another measure of how lakes are doing. So they have indices developed around fish, developed around plant communities. And those are great ways of giving us more texture and more of a picture of what's happening in a lake than just clarity or algae blooms or something like that. Zooplankton provide a couple advantages. One, they're relatively easy to get. It's easy to go out, take a tow, bring it back to the lab and ID what you find. And 2, they have really short generation times and they're extraordinarily responsive to change in lakes. And so I'm hoping zooplankton as a biological indicator could give us a faster sense of change in lakes than maybe macrophytes or fish could. And so we're hoping to partner with the WAF program to come up with one of these indexes for zooplankton. All right. Okay, doing okay. I was a little nervous there about 5 minutes ago, but we're moving well. So I should have trusted myself. I was like right on the dot when I practiced. Anyway, so to talk about my long-term vision a little bit, I wanted to come back to a slide that I put together when I interviewed here almost exactly 2 years ago. So the title of my talk then was Connecting Lake Ecosystem Processes and Services to the People that Benefit from Them. I think this talk in different words is very similar. And I think one thing that I am still very excited about is how good of a fit what I want to do is to FWCB as a program. If you look on the FWC webpage, one of the quotes that I like the most that's there is our research is responsive. FWC is plugged into research problems in Minnesota and research problems more broadly across the world. And it provides a responsive research perspective to provide data to some of these pressing problems that we're facing as human beings. I also talked a little bit about some of the connections I wanted to make in my time here at the U. And I was really excited that I was able to check some of these boxes. It doesn't always line up this perfectly. I guess it's been a year and a half, so it should probably line up. But I have some relationships going with the DNR. I have some relationships going with the PCA. We're using Sentinel Lakes data. I'm plugged into the Minneapolis-St. Paul LTER community. I'm thinking about ways that we can partner on projects here in the Twin Cities. I'm thinking about ways that we can bridge the MSP site with the North Temperate Lakes site, where I spent 9 years. Cross-site work is really important in LTER. It's part of making sure LTER funding is sustained in the long term, is to bring all of these different systems together. And I also talked a lot about sort of this interdisciplinary collaboration and trying to build relationships here at the U. I'll be applying to the IONE Fellows Program. We'll see how that goes in the next year. But I think there's also still some opportunities here that I'm excited to follow up on. And then thinking more broadly of engaging the public and bringing the public into FWCB more. That's something that FWCB does well. I've gotten excited about connecting with people FWCB partners with already, like MACERC and the DNR. Excited to bring my own network into that as well. I also mentioned tribes in this as well. And that's something where I've been working on building those relationships just through phone calls at this point. Now that I finally have some projects and something to offer in terms of a research vision, I'll be going out and visiting with tribes, talking about the projects I have going. More importantly, listening to what they're up to, what their needs are, and see if there's anything I can offer with the projects I have going or anything we could come together with to increase capacity on a research problem. Okay, so just to wrap up, what is lake health? Well, we talked about how lake health is grounded in ecological state. Lakes that are close to their natural state, we consider healthy. It's grounded in socio-ecological contexts. Lakes that provide a range of robust services are considered healthy. And we also talked about how there are frameworks where we can bring in the public or community members and iterate over these questions through time. And that's something I would be really excited to keep working on and keep trying to refine. How can we take this from a lake management framework to more of a lake management and research framework? And then again, I'm not going to get into all of these details again, but the way I go about this is through holistic understanding, making a system as complex and unwieldy as possible, and then finding the experts through interdisciplinary research who can help me tackle it. And then building these partnerships and investing in these partnerships. Like, I know I talked about this individualized medicine thing, which I think is really powerful, and I think it could be a very powerful tool, but it's also just what I like to do. I like people. I like working with people. I like feeling like I'm helping solve a real problem. I get stuck in basic science land, although I like it sometimes. This is fantastic, and I get really excited about this. So I'll leave you with that. Say thanks for your time. Apologize, there's like a minute left for questions, but 2 minutes. So yeah, thank you. Okay. Yeah.
The public idea of clear water is good water. And I just think this acid rain times that my face, they're like beautiful, or zebra mussels. I mean, many areas of gorgeous now. I grew up in your toes when you're up here.
Yeah.
Approach that.
No, in Madison, there was rumors about the diving. Oh yeah, sorry. For those online, the question is, how do you convince the public that something like Clarity is always healthy all the time? And then I think, yeah, so in Madison, there's rumors going around that like the diving community really wanted zebra mussels. I don't think they actually introduced zebra mussels, but that was just something that was always talked about. And I think it does take a fair amount of nuanced outreach. And I think one area where that is going really, really well is in talking about aquatic plant management and wetland management. And I think we could draw a lot of parallels from that. Wisconsin in particular is doing a fantastic job of talking about how you might not want to always be out there killing plants all the time. That's really important. Fish habitat, that's really important for waterfowl. And we want to make sure we're remembering those connections when we manage that. And so I'm hoping to connect with them once we have our ducks in a row here for Minnesota data to start investigating that question, and get out there with the outreach. We have Dan and I have a partnership with some researchers there in Madison, thinking about that more holistic question of aquatic plant management. Of we're managing plants, yeah, but if we're managing plants, we're also managing fish and we're managing waterfowl, and we need to be thinking about all these things holistically. And I think we're on the research side of iterating on that cycle, where it's like we need some data and some stories to bring back to the public and iterate through that. Yeah.
Oh, we'll entertain one more quick question. There's nothing going on.
Yeah, for the invasive adaptive management project.
Mhm.
You mentioned you found that like the contractors are interested in it, so I'm
Yeah.
curious to hear more. In my experience, many groups aren't that interested in evaluating the evidence of things that they do, especially in doing them. They get paid. They might not care.
Right.
How effective it is.
Yeah.
What has been your experience in that building partnerships who are willing to evaluate?
With most of the contractors I worked with when I was at the DNR, for aquatic plant management in particular, invasive aquatic plant management, I was very impressed at how often their goal was consistently like a healthy lake. It was trying to think more holistically. I think there are a few contractors who are very receptive to that messaging. I also think there is something baked in of eventually they want to be able to show results. And aquatic plant management, like any lake management, is super dynamic, it's super variable. And so if they can come in and be like, okay, we're doing well here, that could be great evidence for showing what they're doing. And I think that's what they are hoping for, is we can come up with some dose response curve that they could present to the broader public. I don't think we'll get a dose response curve, but I think we will get some effects, and we will get some areas where we could partner and try to improve our understanding and be more targeted in our approach. But I think, long story short, it's a community I'm really impressed with. And I wonder if that's just, I'm always impressed with people who really think about aquatic plants. I think it's a really interesting group of people who have a strong ecological foundation, and conversations have always been pretty straightforward with them. Yeah.
Thank you, Jake, and I'm sure to be glad to answer additional questions after the summer.
Absolutely. Thanks, everybody. Thank you.